actuation. And now we're working also on artificial flexible robots for medical applications. We just published an article where we show we can dissolve kidney stones with wireless robots. So we're going towards clinical applications. But in fact, the first microsperms were the natural ones, yeah, and we can learn a lot from them. And during my PhD, I was working with sperm cells, but then realized I don't really know much about sperm cells. I mean, we use them for propulsion, but there's so much more to them. So we can learn how do they interact with each other, how do they adapt to different environments, different fluidic conditions, and so on. So yeah, so just as a quick view on what we can do with integrating sperm cells into microrobotics. So we can use them as efficient propulsion source, for example, for these microtubes. Similar here, where we are able to trigger sperm cells by releasing something from these tubes, or we can actuate non-motile cells when we attach particles and then attach rotation. But like I said, this is something we usually, or that's a big part of our work. But we also simply study sperm cells. We observe them. We want to learn from them to improve our design in robotics. So we know that sperm cells are quite energy efficient, but we are also curious about the adaptability, the collaboration among them, so that we can also create robots that can collaborate, that are efficient, that can maybe increase their payload, self-assemble, and so on. And I don't know if you know, but the sperm cells are the most diverse cells. If you look into any type of cells in our body or anywhere, you will see that the sperm cells have a crazy diversity in nature. Yeah, you see here from different species. For example, the fruit fly has sperm cells that looks like a big pile of spaghetti here. Some are not even motile. Some have these hooks on the front or these corkscrews. So it's a huge diversity. And that is because they are under selection pressure. So they are under the competition and the pressure to be the first at the egg and to be the best at migrating through the female reproductive tract. So that has led to this extreme variation. And after my PhD, when I realized, okay, I don't really know much about sperm cells, I decided to do postdoc in applied zoology in TU Dresden, and I got to work with all kinds of species and look at their sperm cells. That was really fun. And here's one example. So you see a fruit fly here, and you see the sperm cell that is stretched out here. So it's actually longer than the organism itself. So it's really crazy. And when you take the reproductive organ here from the male, open up, and you look at the sperm cell, you can see a lot of movement, but not single cells moving. It's more just a huge agglomeration. And when you take a closer look, you can see that these form, for example, these circular patterns. This is what I saw under the microscope, but people have also reported this to see this in vivo. So it's a completely different movement. And I just want to show this here to inspire you that there's much more out there than we think, sometimes in terms of motion, in terms of active motion. It's a big challenge for biologists because it's hard to quantify this motion. So there's one thing where we, for example, apply some machine learning and some other methods to say, how can we quantify this? Because this is a very important model organism, Drosophila. And yet we have no way to quantify the sperm motility. When we look at mammalian sperm, it's a bit more straightforward. So you might see, you might know this structure. This is sort of common in a mammalian sperm, where we have extreme compartmentalization of structure and function. This is also specific to sperm cells. So we have the head, it has the archosomal area that will interact with the egg for the fusion. We have the nucleus here that carries the genetic information. Then we have this midpiece that is rather stiff and has mitochondria wrapped around it, which is important for the ATP generation. And then we have the long tail principle in peace and distal end, that when we look inside, we have this typical 9+2 microtubuli arrangement that gives us the motion of the flagellum. The thing is now that we have this basic structure, but the truth is we have a huge variation here as well, even within one sperm population. And when we look at the morphology, again, we have a big variation. And especially in human, the problem is that sperm quality is decreasing globally. We can see this even by the World Health Organization that keeps correcting its thresholds for sperm health, for example. Now, even 4% of normal sperm is considered healthy. That's our low threshold, yes? So we have a very big, let's say, heterogeneous population of sperm cells. And so the question is, how do we select now, looking at this, for the best sperm? Traditionally, we just look at motility, but it's not the whole truth. And when we look at nature, we know that there are biological guidance mechanisms that somehow select for the best sperm or the healthiest sperm. It's not only based on motility. And we know that sperm cells can respond to a variety of taxis mechanisms. So they can be guided by these stimuli response, let's say. So they can react to chemical gradients. They can react to temperature gradients, pH gradients. So these are all things that have been shown in different species to be present. The sperm cells can also swim against fluid flow, or can swim along surfaces very well. So we can use these mechanisms to select sperm, but we also know that in nature, this is an essential aid for getting to the oocyte. Another question is, so when we look at the sperm migration, why is it that one sperm that makes it and not so many other millions? Yeah, when we look at the sperm migration, the arrival time, at the different locations in the female reproductive tract and how many sperm are left, it's incredible, yeah? So how many are selected to get through the different locations? This is due to the morphology of the female reproductive tract and so on. But why is it that one sperm that makes it and not the many millions of other sperm? And another problematic for reproductive medicine and also for biophysics in a way is that we are used to looking at sperm cells in vitro in a very different environment from what it's like in nature. So typically when we work with sperm cells in the lab, we have a low viscosity medium with a certain composition. We put the sperm cells on a glass surface with a bright light and atmospheric pressure of oxygen. Whereas in vivo, we know that we have very complex medium, that's often highly viscous and elastic. We have boundaries, we have channels, we have other cells. And when we look here at some videos we took, where we have here sperm in medium, then in sperm in a bit more viscous medium, and then here in isolated oviduct fluid, we can already tell that they swim slightly different. So it's very important to think about our experimental design and how we can really include those guidance mechanisms that might give us information of how sperm cells work and how they behave in different environments. So a while ago, we were using a three-dimensional confinement to look at sperm motion, simply here with these roll-up tubes. This was a big thing we did in Oliver Schmidt's lab. We would ingest and roll up all kinds of things and use them for many applications. Here, we would have a sperm cell swimming in this confinement of the tube. We would see that they swim much straighter and faster, actually, than when the sperm cell comes out and wiggles over the surface. So that was already one hint that these surface interactions play a big role. Another thing that I mentioned here is the interaction with other cells. Yeah, we know, we've seen in literature that there's this collective behavior we can see, or even the vortex formation in sperm cells. But another thing we often see is this this bundling of sperm. And we really, we see this quite a lot in bull sperm as well, but it's not studied at all in mammalian sperm. And in fact, when I showed this to a doctor, they say, oh, when we see this, we don't want to see this usually. We want to have nicely single sperm. That's what we want to work with, because it's better for selection and so on. But we see it quite frequently, so we said, we want to look into this. Why does this happen? Under which conditions? Is there any relevance to this? Is there some collaborative behavior that is meaningful? So, and when we look in the animal kingdom, we see actually that this cooperative behavior, or this bundling, is seen in many species, especially in rodents that have these hook-shaped heads. They form these trains, actually only with sperm that are genetically close. So there's some interesting thing happening. In opossum sperm, we often see these pairs, or sometimes we have large bundles of sperm that synchronously kind of crawl through the reproductive tract more than actually swimming. And the evolutionary biologists, they believe that this is a result of sperm competition, and that it's beneficial in swimming over long distances. So there's some sort of energy saving, helping each other out, but there's not much proof for that. So what does it mean, sperm competition? Sperm from different males, they compete for fertilization. And this is known as to be a very strong evolutionary force that leads to adaptations in the design of the sperm, or let's say the way the sperm are built, or they are sexually selected. One example would be longer sperm, that are, for example, moving faster and are therefore beneficial, or these hooked heads that we find in rodent sperm that are beneficial in getting to the egg, or formation of aggregates, where again the geometry of the head plays a role that allows this aggregation. And this all is meant to increase the success rate of fertilization and the competitiveness. Now let's look at bundling in mammalian sperm. And so we started looking at different bundles, and first looking at how many cells usually aggregate. So we see here 2 cells, so they are in pairs. Sometimes they are here in 3 cells together, or here even more. These videos are slowed down, okay, so just so we can see the flagella a bit easier. But we wondered, is this a physical phenomena? Are there biochemical factors? Are there things on the membrane of the sperm that cause this, and so on? So it's a quite complex thing to study. So we could try to break it down. And first, when we look at the number of cells in the bundle, we see that over 80% of the bundles are formed by 2 cells only. Okay? This is simply by observation of hundreds and thousands of these. And then we looked at, okay, how do they form the bundle and how do they start swimming together? And we saw that there's a certain process that's happened. So when we have 2 cells, they, first of all, of course, they have to collide, they have to meet. And these are bull sperm, so they have quite flat heads, okay, so kind of like paddle shape. So they can overlap, that's what happens next. And then there's a bit of a rearrangement so that the heads overlap and the tails point in the same direction. And there needs to be some adhesion on the head regions. So there's a chemical or a membrane factor there. And then they start swimming, they have a transition, and then they start synchronizing their heads, which can lead to actually faster forward motion in certain conditions. So the bundling is a combination of hydrodynamic force and adhesive force between the heads. And so here's the video of one of those transitions where we have the cells, they meet, they adhere to each other, they overlap on the heads, and then eventually they start moving and adjusting their flagella parameters as well. And when we look at the flagella parameters here, we can see that the flagella curvature kind of unifies, the amplitude is more or less the same, wavelength also stays the same. And after a while, they also adjust their frequency to beat synchronously, and that can lead to a slight velocity increase. Now, one thing we notice is that velocity increase is not always observed. So we try to understand why is that, or under which conditions do we really get faster bundles than single sperm, because that's really the advantage that we're looking for. So we tested all kinds of conditions. We started with doing swim up or no swim up. So that's where we have a pellet of cells, and then we have clean medium over top, and then we let this sit so that all the motile cells will go to the top. And when we only look at the top fraction, this is the swim up fraction, we do see that in the swim up fraction there are more bundles observed, simply because these cells are more motile. Yeah, so they have more chance to collide, more chance to form bundles. When we increase the viscosity by adding methylcellulose, we don't really see an increase in bundle formation. So that is more of a collision effect, we believe, that the bundle formation itself. Then we added albumin, which probably, so it modifies the membrane a bit and probably interacts or interferes with the adhesion so that we actually see less bundle forming. We also noticed that over the time, observation time, we see more bundles after a while. So after about 2-3 hours, again, has to do with the chance of them to interact with each other. And then after the motility or viability of the sperm decreases, of course, we also see less bundles forming. Another thing we checked for is the maturation state of the cell, because we know as the cells mature, they have some changes in their membranes. They shed some proteins, they rearrange their surface, we actually changed some charges. So we can use a fluorescent stain to categorize the maturation state of the cells. So non-capacitated would be this full stain, then the B pattern means they're capacitated, so they are already ready to fertilize. Or acrosome reacted, that would be already basically the sperm is done, is not able to fertilize anymore because it's already has released the enzymes from the acrosome. And we see that in most of the bundles, we have actually non-mature sperm, so the F pattern. And when we look in literature to understand, okay, why could that be, is there are some papers that claim that this whole idea of collaboration is that basically you have the long marathon of sperm migration, they help each other out when they're not mature, but once they are ready to mature, ready to fertilize, they single out again, everybody's on their own again. So that's what the evolutionary biologists claim. I don't know if we really have proof for that, but it could be a hint towards that. What about the fluids? Viscosity and elasticity is quite complex, yeah, because it's hard to really have either elastic or either viscous or Newtonian fluid. Maybe for theory it's a bit easier to do that in a model, but in experiments it's a bit harder. But in literature there are some suggestions how to mimic these reproductive fluids in vitro. So we can use methylcellulose, we can use polyacrylamide, mucin, there's a few suggestions. So we did that. And my student Caitlin, she was recording many cells in these different media and looking at the velocity. So here we have the average velocity of single and bundle sperm in these different media. And one interesting thing is here that in the polyacrylamide, which is a fairly elastic medium, we can really see big increase of the average velocity of the bundles compared to the single cells. Okay, so we need to do a little bit more analysis and look into that, but here it's just a brief calculation of the damping factor of these different fluids. And we definitely believe that there's some relation or some effect from these media. So in this, the viscosity itself does not play a big role here, but the elastic factor seems to really make a difference. Okay. All right, so overall, the observations that we have from looking at these bundles is that we believe this is a result of hydrodynamic and adhesive interactions, that the viscosity doesn't affect the bundle speed much, but the elastic media, that the swimming time increases the bundle occurrence, and that the bundle sperm also show higher longevity in terms of their motion. So this is something that we're, I don't have the data shown here yet, but we do see that they basically help each other out to go farther than they would go alone. And this is quite interesting for us as we design microrobots, because we want to see how we can save energy over long periods of time, especially for microrobots. We have to be very energy efficient. Also, how can we move very well in viscous and elastic conditions? How can we increase the payload, dynamically assemble or disassemble, and do multitasking? So that brings me to the end of my talk and key takeaways here. I wanted to show you that there is collaborative motion that we observe in sperm cells that is related to hydrodynamic and membrane surface interactions, and that this cooperation is key, especially in competitive environments. But there's still many open questions, and I would like to extend this invitation also to investigate this also from a biophysics point of view. And if you don't remember anything from my talk, just take away that even in competition, it's good to collaborate with each other. So with that, I thank you for your attention, and thanks again for the invitation.